very privileged to have such wonderful keynote lecturers. And the next lecturer is George Weinstock from the Jackson Laboratory for Genomic Medicine in the US. As many of you know, George has been a seminal role in the Human Genome Project, and before that the sequencing of several entire bacterial genomes, and now he's heading the major microbiome initiative, and we are looking forward to hear your talk. Thank you very much.
Well, thank you very much for inviting me here, and I'm completely blown away by this place. This is just amazing. This is going to be a great conference. Let me find my talk. Might be this one.
Yes.
There we go. Okay. There happened to the pointer. Here it is. I got it.
Turn it on. All right.
Okay.
This is the pointer with the red mark.
Okay. So they always have the microbiome talk about the gut and stool samples and things before you eat. So I apologize for that, but that's just, it always happens. And you were introduced the idea that the Dead Sea wasn't dead, that it was full of microorganisms, and so that's what we're going to start talking about here. And us microbiologists are very humble people, so I call this the human microbiome a new frontier that might just affect everything, because it's true. Okay. So what is the microbiome? First of all, this is a field that is really captured the imagination, I think, of society, more so than some of the other big projects that I worked on, like like the Human Genome Project or things like that. So here's New York Times Sunday Magazine has had many articles about the microbiome in it. The Economist, many of the popular magazines have. These have sort of become the journals for publishing a lot of microbiome research, in fact, because a lot of people use that. We have a microbiome diet now. I don't recommend it, but nevertheless, you know you've arrived when they have a diet. And then there's all kinds of books about... Now you're telling me? Right? Now I'm telling you. The Good God, how to... Microbiome solution for this, microbiome
Please take
solution
this
for that.
so they can hear you in the...
Okay.
Here and that's here.
How's that? Okay? All right. So anyway, it's everywhere. It's everywhere. And just a little bit of definition for the non-microbiologist. This term microbiome was originally coined by Josh Lederberg, a Nobel Prize-winning microbiologist, and this was only in 2001, so this is a very new field. And Josh called it the ecological communities of microorganisms that share our body, and this is the key thing here, ecological. He sort of saw us like a biosphere, just like in macroecology, we have rainforests, we have deserts, we have tundra, we have all these different ecological systems. He saw the body as having many micro-ecological systems, and he called those the microbiomes. And there's another term that's often used, metagenome, which was coined by Joe Handelsman, who's a professor at the University of Wisconsin, the collective genomes of the microbiota. So if you have like in your saliva, if you have the microorganisms there, that's a microbiome, and all of the genomes of those microorganisms there together is the metagenome there. And so sometimes people say you have two genomes. You have your human genome and you have your metagenome, meaning all the microbial genomes. And the point about this is bacteria, microorganisms are phenomenal chemists. They can do anything. They can live on rocks. They can live in 0 degrees. They can live in boiling water. They can live a mile beneath the surface. They can live in the Dead Sea. They can live everywhere because they're phenomenal chemists. They figure out how to get nutrients and energy out of anything. And so because we have all these microorganisms, evolution has selected us to have all this additional biochemical capability by carrying our microbes around with us. It's probably one of the reasons that we're very successful on the planet, is because we don't have to code everything in our own genes. We've got a lot of microbes to help us too. Okay, and so what do we actually have? There's, if you count the number of human cells, people say you might have 10 to the 13th human cells. And if they count the number of bacterial cells, they think you have 10 times more bacterial cells. So you're all just big microbial communities out there, and your body is just a nice environment for your microbes. That's all. You just look at your neighbor, just look at all those microbes. And it's even worse if you look at the virome, the viruses, which are part of the microbiome. There's even 10 times more of those. So you're loaded with viruses, even though you think you're not infected. You're just loaded with bacteria, viruses, everything. That's just the way it is. So, and if you do the math and figure out genes, you've got 20,000 or so genes in your human genome, and you have millions of these microbial genomes. So the gene activity that's going on in your bodies right now is largely, it's just overwhelmingly microbial action going on there. All that chemistry that's going on, all that's doing that stuff for you. So the whole field of microbiome research is, oh my gosh, we have all of these things going on. What's it doing for us? How does it work? And it's really only, the reason it's a new field is it's really only in the last 15 years or so that technology had allowed us to look at such a huge number of microorganisms in the detail that we need to in order to ask those questions. So that's what the driver has been. So here's the saliva, the spit microbiome. These are all the different genera of bacteria that are present. And of course, each one of these, Porphyromonas, that genus, has many species associated with it. So there's actually 5 or 10 times as many taxa in a saliva. And this is what a community looks like. And the size of the font is the relative abundance of the organism. So there's a huge dynamic range. And so our challenge in studying the microbiome is describing these communities, knowing what are the properties of those microorganisms, and then putting that together with what it's doing for your body and what your clinical situation is or whatever it is. And the other part of the challenge is most of these have never been cultured. There's something called the Great Plate anomaly, which means that if you take a sample and you look in the microscope and you count how many microorganisms are there, you get one number, and if you take it and you put it on a petri dish and you grow colonies, you get 1% of these things able to be cultured. So most of these are alive, but they're so highly adapted to their environment, to whether it's the nutrients or the other members of the community, that if you try to culture them by themselves, they don't grow. So the way we know about these is by various DNA sequencing techniques, which I'll talk about in just a second. But this is all culture-independent analysis of a community, and that's the technological breakthrough that allowed this to happen. Okay, and here are different parts of the body. So each one of these pie charts, the different colors are different genus of bacteria, and you can see that the structure of the communities is different in different parts of the bodies. Some parts of the body have oxygen, some don't. Some are nutrient rich, some are nutrient poor, some are oily, some are dry. All those different kinds, like the rainforest or the desert or the tundra, they all have different communities of plants and animals, and so different parts of your body have different communities of microorganisms, and our task is to describe that and relate it functionally to what's going on in your body. And so this is how those experiments are done. Here's a sample. Our favorite sample is a stool sample because most of your microbes are in your gut. And one type of sequencing that is done, DNA sequencing that's done, is to focus on this gene, 16S ribosomal RNA. All bacteria have it. It's a bacterial gene. And it's the basis for classifying bacteria. All those names of genus and species is because of the particular DNA sequence of their 16S gene. that's just the way bacterial taxonomy is done. So when you just focus on just sequencing this gene in a sample, you're doing a census and you find the taxa and their relative abundances. And here's a data point for the mathematically inclined. It's a table that lists what are the different organisms and then the relative amounts of those that you find in a sample. And so all the data mining, all the computational stuff, all the other things that I won't talk about, that's the input, that kind of a table. And the other type of DNA sequencing is called shotgun sequencing. Here you just take all the nucleic acids, whether it's DNA or RNA, and you shred it into little pieces and sequence all those little pieces, and you get some massive number of DNA sequences, 100 million for a sample, and that's then what you compute on in order to try to figure out what's the structure. And the difference here is this is only, 16S is only looking at bacteria, and this is looking at not just the bacteria, but the viruses, the fungi, the yeast, the protist, all the other things. And you can convert those sequences not into different organisms, but into the enzymes, focus on genes of interest, biochemical pathways, all that other kind of stuff. So you get much, much more information out of shotgun sequencing. Anyway, those are the two main techniques. That's the game that we play in order to analyze these. And here's sort of the question that comes out. Here's an experiment. So we always have group A versus group B, group X versus group Y, young versus old, healthy versus sick, this diet versus that diet, whatever it is. And you see a difference in the behavior of the human, the mouse, whatever it is. And you see a difference in the microbiome, whether it's a stool sample, saliva, whatever it is. And in some cases, they may not be causative like they are down here. It just may be this difference affects the animal and it affects the microbiome in parallel. And this could be useful as a diagnostic, but this is what we talk about as correlation. On the other hand, if we do the comparison, and the difference between these two is a difference in the microbiome structure, and it's that difference in the microbiome structure that makes the person or the animal healthy or sick or whatever it is, then there's a causal relationship between the two. And so we're always trying to find causal relationships, and we're always very cautious when we see a difference in the microbiome and the phenotype, that they may just be correlated, they may not be causal. So that's a scientific challenge that we all have. Okay, so I'll give you just a couple of stories. Here's a story, when it works, this is how it works, okay? So here we're studying Clostridium difficile. It's a nasty pathogen, the cause of many, many hospital-acquired infections, very difficult to treat. So, and causes things like diarrhea, it can be fatal. So we want to know, is it the microbiome that's involved in controlling this? Can you do something about this kind of infection? Because these are often antibiotic resistant, so you can't treat them with antibiotics. Okay, so we have a mouse model. This is very kind. The mouse is sort of the workhorse test animal for microbiome. We have one type of mouse that's resistant to Clostridium difficile infection and one type of mouse that's sensitive. And the first thing that we show is that what makes this mouse resistant is its microbiome, because we can take feces from this mouse and feed them to this mouse and now replace its microbiome with this guy's microbiome, and now this becomes resistant. Okay, so that makes us think there's something missing in the sensitive mouse in its microbiome, and we can add that back. It's like taking a probiotic, only, you know, in this case, you're eating feces. And actually, fecal transplant for treatment of Clostridium difficile infections in humans is now the established, accepted treatment when the antibiotics fail, which they often do. And people who have Clostridium difficile infections have chronic diarrhea, and it's a terrible condition, and so they're desperate. And so, yes, they would eat somebody else's, some healthy poop in order to make themselves better. And actually, treatment of diseases by giving stool from a healthy person to a sick person has been going on for centuries. You know, it's sort of like what the witch doctor said. You know, somebody's sick in the village and go to the next door neighbor who's healthy, eat some of their poop, it'll make you better, you know. And a lot of times it works. So, okay, so once we've established it's the microbiome, I told you, this is a before lunch seminar, you know, just what you want to hear, right? Okay. So the next thing we want to do is figure out what is it in this microbiome, in the resistant microbiome, that helps here. So we compare them. We do that 16S sequencing or shotgun sequencing. We compare them, and we try to find organisms of interest that differ between those two, present in this resistant one, absent in the sensitive one. And then once we've identified those, what we want to now is, for those individual organisms, can we transfer just those organisms, not a fecal sample, and cure the disease? So I'll show you how that works. So in this experiment, each one of these rows of colors is a different animal, different mouse. And the mice in these bottom rows are the ones that are sensitive to infection. And the mice in these top rows are the ones that are resistant to infection. And the mice in these rows are sensitive ones that got a fecal transplant from a resistant one. And then down here, these are 50 or 60 different types of bacteria that we're testing for. And the colors, if it's a blue color, it means that particular bacteria is very low abundance. So here's a particular one and it's dark blue. It means there's very little of it in this. And if it's a yellow, reddish color, it means there's a lot of it. So these bacteria, there's a lot in all of these different animals. So what we're looking for now is a bacteria that's rare in the sensitive mice and common in the resistant or in the mice that got a transplant. And without having to stare through this, you can see right here the two genera that are in red here. You can see that they're rare in the sensitive mice and they're abundant in the other ones. So those are the candidates to see, can they cure the disease instead of just using all feces. So here's the experiment then. Here are the species of these bacteria. One of them, this is Bacteroides and Parabacteroides are the genera. And here's the mouse experiment. Here's a mouse that, this is the survival after being challenged with Clostridium difficile. And here's a mouse that got a fecal transplant. Here's a mouse that didn't get a fecal transplant. They all die. And now here we're trying one by one each of the species of bacteria of these genera. And as you can see, nothing worked. So we're experimental scientists. We expect our experiments to fail. Not a big deal. We'll try it again, try something different. And what we tried pairs of them, tried 3. Finally, a combination of 4 of these, when we do it together, works. So here's the fecal transplant, it's protected. Here's the don't do anything, the mice die. And here's a mixture of 4 of those bacteria, and they protect almost as well as that. Okay, so that's the way this research is supposed to work. We've now defined what are the relevant bacteria. You have a very nice, well-defined system, and now you can try to figure out what is it that those bacteria are doing to protect against Clostridium difficile. And ultimately, you might like to find some particular small molecule or something they're making that is toxic to Clostridium difficile or that is turning on the host defenses to protect against or whatever it is. But that's what we do. All right. That's a disease one. What about the other side? What's a healthy microbiome? Is there like a pill we could take with just the right bacteria in it and it would just make us great, like we're 18 years old and athletic and all kinds of good things? So we did the athlete microbiome project. So these are mountain bike racers. These are sort of crazy people who every weekend ride their bikes down mountains as fast as they can and race against each other. And there's sort of 2 categories of people that are doing this every weekend. There's the elite group, the Olympic grade athletes, and then there's the weekend warriors. They're very fit people and they like to race and everything, but they'll never beat the elite athletes. And so we did stool samples from a bunch of these people. Their diets may be a little different from each other, but they basically are on the seafood diet. They're working out all the time. They're so hungry, if they see food, they eat it. So there's no particular thing. Okay. And these colors here are the top 20 or so bacteria found in the stool samples, and each one of these rows is a different athlete, speed play, Yeti, Trek. These are the names of their bikes, not the names of them. So that's how we can track them. But here's the elite athletes, and the way you know these are the elite ones is this is how much they train. And so red and brown is training an awful lot. Yellow is training okay, and then here are the weekend warriors who train less. So obviously the microbiomes in the stool of the athletes are different than these guys. There's a particular bacteria here that they have a lot of that these don't. This is the bacteria called Prevotella. So they're different. They're all healthy people, but the elite athletes have something else going for them, and it has to do with their gut. We haven't done fecal transplants. We haven't put Prevotella in a pill that we could all take that would make us young and athletic and everything, but it's coming. Then there's another bacteria that we found that you don't pick up with 16S analysis, you do other ways. In Mira's slide, it was mentioned that in the Dead Sea, you find a lot of archaebacteria. So there's 3 kingdoms of life on the earth, right? There's bacteria, there's eukaryotes, which are animals and plants, and then there's archaebacteria, which are these strange things that can live in places like the Dead Sea and other places like that. And they have a different kind of metabolic setup than the other bacteria do. But here in the athletes, here's a typical athlete. Here's the archaebacteria that we find. This is like a pie chart with all those different types of bacteria that are there. So only about 1% is this particular archaebacteria. Here is the transcriptional activity going on in that person. Half of the transcription, half of the gene expression going on in their gut is from that particular bacteria. So it's not only that they have it, but that it's going wild in the gut to do something. So we have a hypothesis that we could test. The metabolism of these archaebacteria, like I say, it's different than bacteria. Here's food and here's the bacteria in your gut, and they have certain well-characterized endpoints of their metabolism. That turns out to be the food that archaebacteria like to eat, things like hydrogen and carbon dioxide and stuff like that, and they end up making methane as their end product. So having this, our hypothesis is having this means out of your food, you get sort of an extra bounce of nutrients and energy and things like that being extracted from the food, and that gives the athlete something extra. So there's something nice that makes sense that we can test. Okay, so there's many things, projects that we have going on that are just doing these kinds of analyses. Necrotizing enterocolites, these are preterm babies in the neonatal intensive care unit. Their guts are immature, they don't have a microbiome yet, they don't have an immune system yet, they're sensitive to all kinds of things. So we study how to manipulate their microbiome to prevent them from getting some of these kinds of infections. Diabetes, of course, is a biggie. Fatty liver disease is a biggie. Multiple sclerosis, we've done experiments to the point with mouse models of multiple sclerosis. You can protect against multiple sclerosis with intermittent fasting. This works in mice, and it's also one of the ways that humans have for dealing with the disease when they have it. Well, if you take stool samples from a mouse that's been fasting and is protected, and you put it into a normal mouse, that mouse is now protected against multiple sclerosis. So it's all due to the microbiome, and we have a bunch of candidate bacteria that we think are there. Sickle cell cocaine addiction, we have a great project going on trying to show correlations between behavior, what's called novelty-seeking behavior, kind of addictive behavior, and the microbiome. And these are in mouse models where you either measure behavioral things that they do that are correlated or actual self-administration of cocaine. And there are correlations with certain bacteria there and that. Anyway, the list is long for many of these things, and we can always find some kind of effect. But like I said in the beginning, it's this challenge, correlative versus causative, that we're always trying to look for. And just where all this is going, we just completed a diabetes study where we not only looked at the microbiome in stool samples and nasal samples, cheek, skin samples, but we looked at all kinds of things of the host, cytokines, the immune response, transcriptional patterns, the DNA sequence of the human genome, metabolites, or you name it, and really to understand what the microbiome is doing mechanistically and put it together with a disease or something, you have to know everything that's going on. So this is a massive data set that poses a lot of sort of state-of-the-art data mining challenges that we're using to address that. Okay, what can this contribute to Dead Sea science? All right, so Mira set this up beautifully by saying that the Dead Sea is not really dead. There's stuff in it. Now, when I kept talking about human diseases or mouse diseases, so we're always doing microbiomes on these two things. There's been so much work on human that's done that if you do 16S sequencing and you get millions of 16S sequences, 95% of them are known. They're in a database. You know what the organism is. There's only a few new things that you discover when you do an experiment. But when you do that for the mouse, only about half of them are known. There's lots of the bacteria in the mouse, because it's less studied, are in the databases and are actually known what they are. And the same thing with shotgun sequencing. For humans, 40% of the shotgun sequences, we don't know the organisms that they're from because their genomes have not been sequenced. And so we get a bunch of sequences, but we don't really know where they're coming from. And it's worse for the mouse. More than 50% of the sequences in mouse are not known. So the challenge is, in order to make sense out of these data, is to try to figure out what are the other organisms that are present so we can relate it back to 16S and shotgun. But like I told you, most of these organisms have never been cultured. Doesn't mean that they're not culturable. They might be culturable, but they just haven't been cultured. So we can't, and there's hundreds of thousands, millions of organisms, so we can't just go out picking colonies and trying to get each one. So we have to live in the world of just culture-independent DNA sequencing and try to figure out what the organisms are and what the structures of their genomes are. So we've been developing a lot of techniques for doing this. For 16S sequencing, we use new sequencing instruments like the one from Pacific Biosciences, PacBio, that generates very long DNA sequences so we can get very accurate species-level definition of what the organisms are there from the 16S. So all of these new organisms with 16S sequences we can do something with. And for shotgun sequencing, there's a couple of different methods that we use, one of which, if you have a bunch of different samples and some organism is in varying amounts in each sample, all the sequences from its genome will change in their abundance together in the different samples. And so, yeah, you may have 100 million sequences from each sample, but if you have a way of figuring out how they change between samples and putting together all the sequences that change coordinately, now you've got the genome, and you can assemble that and make a genome without doing anything. Computationally, it uses a canopy algorithm, which is actually something that has been in computer science for a while for other purposes, but applied to this, it works very, very well. We discover hundreds of new genome sequences without culturing anything. And then there's other technical things you can do using a technique called Hi-C. So anyway, my thought is the Dead Sea is loaded with things, and we don't even know half the things that are there because they can't be cultured and stuff. Sounds like a good application for some of these methods where all you need to do is collect samples, sequence them, and then do some fancy computational stuff. And now you start to find genomes and organisms and their functions. And remember, these are the greatest chemists on the planet, and you might find all kinds of very useful things there. Okay, so I will stop there. We have a great group of people working on these things, great support from many different places. For the medical things, where our hats are off to the subjects and their families who we work with. Thanks very much. Now we can eat.
First of all, some questions. Thank you very much for this very exciting talk. Please.
I have a quick question. Actually, I have two quick questions. One of them has to do with
this issue about the 16S ribosome versus the 18S, which was used to differentiate the eukaryotic from the prokaryotic, or the bacterial and archaeal sequencing. You're not looking into the 18S as part of your... Okay, so let me ask you a quick follow-on question. So I was very interested in wound healing for a long time, and I did a 16S analysis of chronic wounds of diabetics versus normal. And so I've been very interested in this issue. And recently, I've become interested in this question of whether or not the microbiome has an influence on behavior. So there's lots of microorganisms that change the behavior of animals. Toxoplasma is a great example where infected rats our brains are infected, and they see their predators like cats as potential mates. So rather than running away from them, they run after them, right? So this is from a specific parasite. Rabies is another example. Do you know any data about whether the microbiome, people who have dogs, for example, completely change their microbiomes, right, significantly when they are living with their dogs, or their boyfriend and girlfriend living together? How does that influence behavior, and what do you know about the behavioral correlates?
It's a great question. Personally, I think it's going to have a huge, deep effect. Practically, it's at a stage right now where there are lots of papers that show some microbiome effect, which is different between people who are stressed or not stressed, or have bipolar disorder or not, or schizophrenia or not. But it goes back to this correlation versus causation. Now we have to get into that. Sort of relevant to this, though, the microorganisms in your body produce neurotransmitters like serotonin, GABA, things like that. In fact, your microorganisms produce more serotonin than your nerve cells. And it doesn't cross the blood-brain barrier, which keeps you from going nuts. But nevertheless, there's a tremendous amount of nervous influence that the microbes can have. And your GI tract has its own nervous system called the enteric nervous system, which is innervated and goes to the brain, and sometimes called your second brain. And that's also where all the microorganisms are. There's got to be some profound thing going on here. Why, you might ask, do microorganisms make so much of a neurotransmitter like serotonin or something? If I was wildly speculating, I would say that as humans evolved, they had a lot of parasites. And microbes were helpful to the host by interfering with parasites' nervous systems, by making a lot of neurotransmitters or things that paralyzed, tapeworms, or did things like that, and that there's some kind of selection going on here.
So, well, thank you. That was a very inspiring talk, and there was a challenge to look for new organisms in the Dead Sea. So since you just look at DNA, you found genes, or whatever, 16S, how do you know that it's bacteria living in the Dead Sea, or just DNA that got there from dead ones?
So just seeing the sequence doesn't tell you that there's something alive doing something. So when we look at viruses, for example, we sequence, we find the viruses that are there. If we look for DNA viruses, and we also do the transcriptional analysis, and we can see genes from DNA viruses that have RNA sequences, we know those viruses are causing an infection. They're not just in a virion, they're not just a piece of meat. So the same thing for bacteria. We find M. smithii is only 1% of that particular individual, but 50% of the transcriptional activity that's going on is M. smithii. And when you look at the genome, it's actually methanogenesis genes. It's a number of different things going on. It's not just like one, locus that's going. So that's the kind of evidence that we have for that. There's also another trick that you can do. If a chromosome is replicating, bacterial chromosomes have an origin of replication and a terminus of replication, and they go bidirectionally. That that means that a growing, replicating chromosome will have twice as much DNA from its origin than it has from its terminus. So with a shotgun sequencing, we can just take that ratio for different organisms, and that only not only tells us that they're growing, it gives us a growth rate. It's amazing to think you can get a growth rate of many organisms in your gut without ever culturing them or looking at it or something like that. So
They may be replicating very slowly.
some are, and some can be dormant. You know, there's all kinds of things. Yeah.
I need to cut because it's so. Thank you very much. And actually, we have a whole session on microbiome right after we will eat and be ready. and you know, and we can even run a little bit before that. So we will cut now, and we have lunch ready for us. We planned it for an hour. We will try to cut it a little bit shorter to compensate on time. And thank you very much for the inspiring talks. Thank you.
So Mira did not give time for addition. I have a question about the intermittent fasting.